Thank you. Right. So my name is Mats Gabrielsen, and I'm just here for two reasons. One of them is to highlight that the CCP4 has an EDI group, and we would very much like to hear from you. And how to do that, I'll get back to in a second. The other thing is to highlight this is the code of conduct for the workshop. You've all ticked the box that you have read this when you joined us or signed up. This is just a brief reminder. The full EDI policy is on that barcode. More importantly, the EDI feedback form is totally anonymous. If you have any concerns, anything you want to address, or you want to be involved further with EDI work within the CSP4, you can fill that out, or you can come and find one of us. Most of us are here this weekend. If not, send us an email, contact us via the form. Enjoy the rest of the, oh yeah, sorry, I should highlight, we do have a website. I really urge you to go and look at that. We have a roadmap for what we are hoping the direction the CCP4 should move in the future. And there is a quiet room here if you feel the need to go and have some space yourself. Go and sit down. And can I also ask that you don't speak to anyone who sits in the quiet room, because that kind of defeats the purpose of it. With all of that, enjoy the rest of the weekend and all the great talks. Thank you.
Yeah, it's on. Okay, let's continue with the panel discussion. And we have a very interesting panel of speakers, somebody who's in the early career stages of their career, somebody from industry, and somebody with a more advanced academic record. Can you please introduce yourselves?
Yeah. Yeah. So my name is Alizia Fadini. I work with Randy Reed at Cambridge University. And I can say a little bit about what I'm interested in. I'm mostly work on trying to find new ways that we can extract information from structural data sets, including AI. So that maybe is relevant to the rest of the workshop.
So I'm Tom Davis. So I'm a protein crystallographer by background, and I've worked for the last 23 years now at Aztecs Pharmaceuticals in Cambridge in the UK. And for those of you who aren't familiar, Aztecs is a drug discovery company which has fragment-based drug discovery, which we've heard quite a bit about, at its heart. And I guess we sort of champion the use of crystallographic screening in particular. So X-ray crystallography is really at the heart of what we do, although increasingly we're also moving into cryo-EM now, as a lot of people.
Okay, so my name is Julie Ménétré. I'm a scientist, a group leader in France at Gif-sur-Yvette in the Institute of the Integrative Cell Biology, close to the Synchrotron Soleil. So I were trained as a crystallographer during my PhD and postdoc too. And now I'm a crystallographer since 20 years. And I'm also make some other things that crystallization and crystallography.
Okay, and I'm Robbie Joosten. I'm at the Netherlands Cancer Institute. I develop methods for structural biology, and I'm playing chair for today. Alicia, you are in the early stages of your career. During your PhD and now with your postdoc, do you think there were any particular gaps in your education at university?
Yeah, well, I guess I can give a perspective that's obviously quite personal. So I was in the UK system, and that's a very specific, usually it's a very specialized type of, I assume we're talking undergrad, maybe leading up to PhD. So, so they're kind of very specialized courses. So I was trained as a structural biologist and biophysicist. And considering that I was coming from this UK education and that I'm primarily interested in developing methods, I can say that maybe what I found a little bit difficult at first was the fact that not having a slightly broader education meant that in the very interdisciplinary field of trying to integrate some more mathematics, some statistical mechanics, and some computer science into new software or new ways of analyzing data, that was something I had to complement with like my own time and new courses. So I think obviously that's also my personal interest that led me to have to maybe shift the focus a bit. But I've also heard from quite a few of my colleagues that are more interested in studying a specific system that they would have liked to have some tools to do a new type of analysis for the data that they had collected. So I think that's something that, again, I know it's quite different in other countries, but at least in the education I was looking at, I was involved in, could have been better.
Okay, and Julie, when you hire PhD students, do you think there's something particularly missing, perhaps in the French education system?
So in the French education, the PhD training is 3 years. Sometimes you can have a grant for 4 years, but it's very short time. And what I see is that it's more or less 50 years that I hire PhD, and at the beginning, and my generation was trained like a chemist, physicist, biochemist, and not really as a biologist. And now when the student apply for a PhD position in my lab, they are mainly biologists. So they in their background, they miss the science computing background, chemist background. Sometimes it's important. We see the previous talk, it's something important too, and physicist also. So this is something really different to work in crystallography.
And Tom, can you give us a bit of an industrial perspective on?
So, I mean, I would agree with the comments that I think in the UK, it's quite a short specialist program for PhD students. And I think particularly within industry, we value scientists who can really see the kind of bigger picture, because I guess particularly in drug discovery, it's intrinsically very interdisciplinary. So if there was the opportunity, say, for students to be, PhD students to be more broadly trained, I think that could definitely be beneficial. And also, you know, things like soft skills as well, team working are also extremely important within industry as well.
And do you, as Aztecs, reach out to universities or do educators to sort of describe your requirements, or?
So actually, we haven't done so much of that. Our interactions with, you know, specifically at Aztecs have tended to be more at the kind of postdoctoral level. I can't really speak for other companies, but I think there's definitely a niche there probably for companies to work at an earlier stage, kind of defining curricula and things like that. I mean, we've talked a bit about, I think it just came up around kind of interactions with chemists in particular, which is something very relevant to industry. And I think having better training of structural biologists in terms of chemistry, for example, is increasingly valuable. So that would be some feedback, for example, in terms of curricula we could give.
And Julie, as an educator, as organizing courses, would you think, would you appreciate that sort of input from industry?
Yes, so I think the last 4 PhD that I trained as a crystallographer now work in a private company. So I always say that it's very important, the interface with academia and industry. So for me, it's a good things that they go into
so there's
private
probably more
company.
room for collaboration there and engagement.
In the same time, there is inputs from industry in my team, but I will appreciate more. I think it missed something for the.
Alicia, so you already mentioned a few things that you think were important for your personal education that you could have had extra at university. Other things that you think are more important in general? Do you think that everybody should know for doing structural biology research?
I think this is pretty obvious maybe, and it's also improved compared to even when I did my undergrad, but I do think like programming, like a solid ground of programming is quite inescapable right now. I don't think just because everyone needs to become like a developer, but just to have the flexibility not to depend on some black box software and have the creativity to do a new type of analysis. So that's one thing. I also think I do work a lot with AI now, and I think AI safety and also interpretation of data that might come from AI will be like an increasingly important, like we've seen this kind of boom now, and I think it's starting to settle, like it's the idea that these models are not perfect and that we have to also be very careful about the conclusions we draw from them is becoming more important. I think it should probably become part of some more formal education.
I mean, maybe I could just second on the programming front. I think I totally agree with that. It should be a standard part of scientists' toolkit, if you like, to be able to operate in the most efficient way. So I think that would be really useful for more broad programming training.
And Julie?
Yes, I agree with that. This part of the training of the structural biologists. So there is more biologists, and they are, in my experience, they are a little bit afraid by mathematics, by science computing, and they don't want to go to be trained with this and miss really this part for the structural biology project.
No,
Yeah, I was just going to say it's also the point about machine learning as well, which is just becoming kind of common, almost kind of general knowledge that I think scientists should also have some kind of background in as well as part of their training, as another part of the toolkit.
Yeah, I just think if you have some understanding of what it is, you're able to better interpret what it is that you're using and the pitfalls. Yeah, exactly.
Yeah.
Now, if you want to like make sure that scientists get additional skills, we probably also have to slash a few things from the curriculum. Now, if you think of a standard like crystallography course, what would you take out then? And I know this is a bit of a mean question, but see where can we skimp a bit so we can have more space for learning about machine learning and AI and maths, and perhaps a bit more chemistry.
Who's going to go first on that? Well, yeah, I mean, this is going to be controversial, but maybe more of an, if you had to cut something, more of an emphasis on molecular replacement rather than heavy atom phasing, perhaps, as things has often moved on from there. So things like heavy atom derivatization perhaps could be something that you could consider maybe having less of. Just recognizing the fact, I guess, molecular replacement is probably the dominant phasing method now. So maybe that would be something.
And Julie?
Yeah. And you need to using molecular replacement now. The model can come from AlphaFold or other software, and you need to have people that know the structure and what is a structure, what is a protein or a molecular biomolecules, and what is a structure and how evaluate the structure if this is good or not, and etc. So this means chemistry knowledge too.
And you've organized the summer school for quite a long time. How have you seen the curriculum of that summer school change over the last 10 years?
Yeah, so I organized two types of workshop. The first one was is a 1 week workshop in crystallography. So we go from the crystallization essay to then the data collection at the synchrotron Soleil. We have 1 day to do that. And then the phasing method, the scaling, the refinement, and then the evaluation of the validation of the structure. So we do that in 1 week. So I found it was short, but the student, the PhD come with their data, and we can help them to solve the structure. And the other one is a 1 week also, but each day we learn, we have course on different structural biology approaches. So there is 1 day for NMR, another one for crystallography, another one for cryo-EM, etc. And in fact, I realized that we try to do in 1 day what we do in the other workshop in 1 week. So the idea is not the same. The idea for the second workshop is to the PhD student to have an idea of what will be all the pipeline to obtain the information, not to be to let the PhD at the end of the workshop autonomous in the different approaches. And the last one, we do that since 14 years. And at the beginning, one more time, we have PhD students that have background in physics, in chemistry, in science, computing. And now we have students with biological background. It's good, it's important, but there is less and less people with chemistry and physics background.
So you mentioned automation as well, and so that's an important point. Do you think, and this is for Tom and Alicia, do you think that automation will, to what extent will automation replace trained structural biologists in an academic setting or in an industrial setting?
So
You
I
go
can make,
ahead,
I make a
yeah.
comment. So most of our automation has been around our software automation in terms of a pipeline which will produce fitted protein ligand complexes in the most reliable way. But I think we still find sufficient edge cases that things don't always work with an automated pipeline, that we need somebody with that sort of in-depth crystallographic knowledge still. And this can actually happen more often than you think. So I think it can happen because you're looking at kind of increasingly complicated systems. But actually, even within what you might imagine would be relatively straightforward cases like different ligand soaks, you can get changes in space group and things like that, or unusual kind of twinning pathologies where you actually need somebody with that depth of experience. So for that reason, we still really highly value crystallographers with that detailed experience, you know, when we recruit, for example. Yeah.
And how do you see that, Alicia?
I mean, I think these points are points that I would foresee being perfectly applicable to academia as well. I think my perspective, at least how I see one aspect of automation, is also that I think automation provides a great opportunity from some perspectives, automating data collection and increasing the throughput of structures that we can obtain, and maybe also improving as much as possible the software that leads us to some interpretable models. Opens a lot of possibility for doing some more statistical analyses of crystallographic or cryo-EM structures and dynamics. So I think obviously all these points are perfectly valid, and I think they probably always will be, but also I wouldn't shy away from automation in the sense that I think it could just be a great tool to open a whole new range of interesting biological questions that can be addressed.
So I think we should be embracing it, as indeed I'm sure everybody is, because it allows you to look at more things, take some of the drudgery out of things, allows people to focus on downstream analysis more, again, at those interfaces with other disciplines like chemistry and biology. We just need to make sure with these automated pipelines that I think the generation that we're training are fluent in protein structure as well, and can really sanity check what is actually coming out at the end of the day. And that goes for protein structure and also, again, for ligand chemistries coming out to make sure they're sensible.
So we've already mentioned courses, and there's quite a lot of courses that are outside of the scope of just university education. There's the CSP4 courses, of course, there's Cold Spring Harbor and Rapid Data. Have you been involved in any of these? Have you been to one of these courses, for instance, or have you taught at any of those?
So I can say I went to Rapid Data actually when I was an undergrad student in California, well, kind of doing a research placement in California. For me, it was very useful. I think because I was at the point where I was actively trying to solve a structure and actively trying to learn ways of going to a beamline and getting to some functional observations at the end of the process. So normally I wouldn't necessarily say that attending during the undergrad is the best way to go. I think probably attending once you are at a point in your training where you're very interested in the practical aspect of collecting data, that's when it's very useful. So I would imagine that if you just taught it to a room of undergraduates who were nowhere near a beamline, you would probably just easily forget some of the details, whereas like the practical aspect. But it was super useful for me.
Yeah, I think it might be a bit too abstract at the undergraduate stage, perhaps. I mean, I attended a course in the first week of my PhD, I remember, not a CCP4 one, but another one in York where I did my PhD. And even at that stage, I remember just being totally overwhelmed by this in the first week and thinking, oh my God, you know, have I done the right thing here? And it took a long time for the information to sink in. But nonetheless, it was ultimately very useful, obviously.
And yeah,
I think it's important
Yeah.
to for the course. Also, I agree with all you said. Also, for the PhD student to set up a network with their teacher, but also all together, because there is a community that is very important for strengthening the skill and the development of the field.
yeah, I think the networking aspect of these courses is very important. You see people appear through different stages of their career that you first met at one of these courses. And I think that's very exciting, and you can see the effect. So obviously these courses are typically very cutting edge, right? Because people are working with their actual crystals and things. Do you think also at later stages in your career, it might be useful to have sort of well, not a group leader version, but like an advanced crystallography course in modern techniques or something? Would that be interesting?
I mean, I think putting it in the context of other techniques is always important, and it comes back to what I was saying about, you know, maybe by my mid-stage career having that much broader picture of how it all fits in with other scientific disciplines as well. So I think there's definitely room for that.
Sure.
And now one other question. So we're talking about mid-career things now. If you had the opportunity to hire the ideal mid-career scientist, either for industry or for academia, what sort of work experience would that person have had? Let's say that you are hiring somebody who's like 10, 12 years after their PhD.
Well, I mean,
hiring
the question
stage,
is more
but-
for
yeah,
you.
I mean, you know, clearly, they will want a firm technical basis, you know, in the discipline. So for crystallography, we would want somebody that was probably experienced in the wet lab from a kind of protein biochemistry point of view, as well as the crystallographic process in terms of solving structures, and then the very important downstream use of those structures. I think definitely want them to have a kind of broad picture, as I said before. So rather than just a narrow vision, a broad picture of where it would all fit together. And also by that stage, you know, experience probably running projects, that side of things, and determining the scientific direction of where they were going to take the research independently, as well as interacting with a kind of bigger team as well.
Julie?
Yes, I think it's important to have a strong expertise in one methodology, because not only the methodology, but it's important because it's mean that the researcher is also able to have a critical thinking, scientific integrity, is able to evaluate the results. It's very important. But in the same time, it's also in the mid-career hiring somebody that will have a general view of all the structural biology. This means that when she or he will have a biological question, what will be the better approach to solve the question? Probably not is her main expertise, but he can know what will be and find people to work with. So both are important.
Yeah, so I think it's that balance between the broad kind of holistic view and then within their own specific domain of expertise, that detailed knowledge as well. If you can marry up those two things, probably.
And Alicia, does that change your career plans, or do you think that in 10 years it will be different anyway?
I think it highlights what I already think is important, which is the interdisciplinary aspect of trying to grow in this field. I think there's an interesting point that you mentioned, which is the ability to work within a bigger team. It's something that I felt quite lucky during my PhD, and even now, because I've worked in these bigger projects that often involve big interdisciplinary teams. I was doing some XFEL experiments that usually won't function unless there's like a set of at least 30 people working. And so maybe that's not the most common academic experience, but I think in general academia, even at the postdoctoral level, is very much a working alone, or not completely, but definitely different from the colleagues I know who are in industry.
Yeah.
And that's something I think that we are maybe missing out on a little bit in academia. I think there could be a lot more interesting projects happening if there was some better way of accounting for merit at the end of a project, like authorship on a paper, and therefore there was some incentive to working together as a team more.
And I think that's one downside of the kind of traditional one PI on the
Yeah.
paper sort of approach. And you know, within industry, by its very nature, you have to be good at working in a team. You know, it's a bit of a kind of management buzzword, but it really is true. You need those good interactions with all the different disciplines within the company, because clearly you're not going to be an expert in all the different things.
So at this stage, thank you all for answering all my silly questions. But also now I want to turn to the audience and ask you, are we training structural biologists for jobs that won't exist? And I'd really like to hear also from the more early career scientists if they feel that we are doing the wrong thing, or are we doing the right thing, or will you just learn on the job anyway? And then afterwards, the more senior people can also weigh in, of course. But is there anybody in the audience that wants to contribute or has a question for the panel members? That's of course also fine.
James Naval, Curve Therapeutics. So I remember when I was getting started in structural biology, and then I heard about AlphaFold, and I've seen these structures come out and got quite scared that I would no longer be needed at some point. Do you think that eventually AI will take over the role of a structural biologist completely?
So, I mean, I can make a comment on that. I mean, you know, there's no doubt, obviously, as people be aware, it's been a massive step forward, AlphaFold, and has really augmented and speeded up, I think, a lot of what we can do. I think we see it as really complementing, you know, what we do at the moment, rather than replacing it. And I suspect it will remain that way probably for the foreseeable future. I'm talking about things like predicting different protein conformations, uncovering kind of cryptic pockets that might only be present when a ligand is present, different pHs and the effect of that on the protein structure. And that's just from a kind of protein point of view. If you think about predicting protein ligand complexes, I mean, obviously there's been a step forward with AlphaFold 3 and similar things. I think models like that need a lot more benchmarking by the community to understand how useful they are in terms of predicting protein ligand poses. But I suspect, if only because there will be a lot more training data needed on things like protein ligand complexes, which will require, you know, experimental structural biologists to produce, we're quite a way off it forming, you know, an entire replacement. Probably the number of experimental structures might go down over time, but I would imagine maybe not drastically. And we'll probably also start just looking at more and more complicated structures as well, more and more protein complexes. Yeah, that would be my thoughts on that, I think.
Yeah, I also think that the AI methods have actually led to more people wanting to know about structural biology. At our home institute, you can clearly see that many more groups are now talking about structures, even if it's just a screenshot of an AlphaFold structure. The idea of putting everything in a structural context, this has become much bigger now also in cell biology. And I think that even if we might lose a few jobs in solving structures, there will be way more positions in interpreting structures.
Yeah, I totally agree with that. Because the structural biologist is probably the person that is most familiar with protein structure and the quality of those structures, and sanity checking again what comes out of methods such as AlphaFold, and interacting with say biologists or chemists who aren't so familiar with protein structure. So you can see maybe the role shifting in that direction towards the interpretation as well.
Yes, over there.
Sam Horrell, Imperial College. So obviously this is a room full of structural biologists, so we know the importance of us in the field, but I'm sure I'm not alone in finding that a lot of funding bodies are now starting to seemingly like reject crystallography projects as, well, AlphaFold has solved this problem. So what can we do in the future to keep crystallography relevant and bringing money into a lab? Because obviously without money, the research can't continue.
So again, I guess it's partly about proving the cases where at the moment perhaps AlphaFold methods don't work as well. So the bigger complexes, different conformations, things with nucleic acids, those liganded structures. Clearly, as the AI methods improve, which I'm sure they will do, you know, the use case for crystallography may come down. But again, I think there are sufficient cases where we will need it for the foreseeable future.
So maybe
yeah.
I have an opinion about this. At least my perspective is that we've obviously, in terms of AI not replacing structural biology, there's one aspect which is, and it's already very obvious to anyone who's trying to develop machine learning models, and that is that if we're interested in the next frontiers, ensembles, dynamic complexes, protein ligands, there's no training data. And so that will obviously need to somehow be produced and analyzed. But I also think, at least I would really love to not just see structural biology as the field that's going to produce the next PDB ligands or PDB ensembles, right? Not just as the production factory of new data that machine learning groups can train on, but I think and it goes back to this interdisciplinary and like training of structural biologists also in the tools that are available, physics-based methods, but which we shouldn't forget about, but machine learning. Or at least I plan on really exploiting these new tools to answer new questions that we don't know how to solve right now. So can we use diffusion models, generative models to start producing some ensembles that we can compare to the data? This is just one example. And so if we open new frontiers for interesting questions that can be addressed by integrating structural biology, cryo crystallography, and machine learning models, I think there's actually quite a lot of interest in that front, and there's definitely interest from the industry perspective, and I would expect there would be from funding bodies as well. So I think it shouldn't just be seeing machine learning as a separate field that might eventually use our data, but it's more, okay, let's us use these new tools, let's become proficient in using them so that we can develop new ones and integrate our data to process them. This is at least what I plan on.
We've been there before where we've seen technologies and this was going to solve this problem and other techniques would all go away. And of course, what's happened in practice is you use all of these techniques in synergy. And I suspect it will just be exactly the same here. Yeah.
David from IO4 Beamline Diamond Light Source. I have two things to throw. They are contradictory, so come from different angles. The first one is, aren't we, considering not structural biology, but micromolecular crystallography, maybe we were a bit spoiled, we were the kings of the mambo. When we got a structure, we got a paper. When we got a structure, we added a lot of information that probably you didn't need to do other techniques. But now with the challenge of AlphaFold, cryo-EM, etc., maybe we're no longer the kings of the mambo, and we need to look more at integrative structural biology. Now, what I would like to throw is, someone in another field was doing AI for looking at analysis of 10 different techniques on the same sample. And then in collaboration with industry, AI could actually summarize the best of the 10 techniques and give a bit of the juice that is needed. But the industry said, no, we don't want that. We want to do 2 techniques, run the AI and tell what are the 2 techniques I need to do to get 90% of the results, but without spending the money and the time of the 10 techniques. So AI can do the analysis of 10 techniques, but that's too much time and too much money. aren't we in this situation where we maybe need to use AI and combine techniques? Sometimes we don't need a structure to take the conclusion. What do you think about this in the panel?
I mean, yeah, I'm not sure if I totally understand the question, but certainly from a drug discovery point of view, I think of course, drugs can be developed without structure, but there are definitely clear advantages in doing so, in terms of speeding things up, in terms of predicting binding modes reliably, predicting affinities reliably as well. So yeah, I would still, I mean, obviously I'm biased, but I would still see a strong use
an AlphaFold
for
model?
structure alongside other computational techniques. You know, I think studies have shown, for example, if you use an AlphaFold model for things like docking or free energy perturbation calculations for affinity prediction, you degrade the signal there. And in some cases, maybe that's okay, but there is still clearly a strong advantage at the moment in terms of using experimental structure.
Yeah, that's also the point that we might have been king of the hill at some point. I mean, I think we still are, but in a different way. It really is the interpretation, and we should perhaps reach out a bit more to other communities and say that, well, we've been doing structures for quite a while, and even though the solving of structures might become less important, the interpretation is really our strong point. And I think that the thing that we're missing now is also the validation of these AI models. And you can do that with other techniques. You don't necessarily have to get the structure, but we definitely need to validate all these predictions because quite a lot of them are, to say at least, a bit overconfident.
That's what was my point, because you could do other techniques. If I can throw so the other question, then I release. The other is the opposite way. You know, 2 decades ago, electronic engineers and electric engineers in UK realized that the students they got at 18 years old arriving to university knew much less than they had 3 or 4 decades ago. So children in their teenage times, they would tinker with things in their garage, they would do amplifiers for their bands, they would solder some circuits, and now this was not happening anymore. And the professors in University of UK had the idea of creating the Raspberry Pi. And for a while this worked, you know, they came this cheap thing that you could think at home. And for a decade, they were able to bring back children at 18, 19 years old to university that already had some experience because they had curiosity. The curiosity was always there, but they were distracted with other things. They were able to bring them back to this. Is there a Raspberry Pi for structural biology? And maybe we need to think about it and interest people, children from the 14 to the 21 back to this.
Yeah, you want to weigh in? Otherwise, I'll advertise a bit that tomorrow we'll be talking about Foldit, which is sort of a Raspberry Pi for structural biology, and it's definitely something that young people can tinker with. And it's I think that children are naturally interested in lots of different things, but you have to make it exciting and you have to beat the competition. And the competition is TikTok, but it's also ChatGPT, and so we have to make something very exciting.
Increasing distractions.
Yeah, you have to get rid of the distractions. Well, if there's another Raspberry Pi, that's all this discarded home sources that we don't use anymore, but that's you can only reach so many people with that.
Minor again. The number of X-ray structures is already going down if you look in PDB. It's already going down, yes, per year. So this is the one thing. The second thing, that we use the term structure solution. Now we should use the term structure determination. And the third thing, we do not train, or at least in my lab, I do not train people to be structural biologists. I train people to be scientists. I train people how to solve problems, any problem. And they are, and if you would look, I had 130 people in my lab, unbelievable large number. Some of them are head of small companies. Some of them have position in computer companies. Some of them created companies. Some of them are working in structural biology, but rather I would say biology in general sense of that. And I think that we still can do unbelievably good job training people to solve problems, because there is a lot of problems to solve.
Because
Yeah.
so the subject of the discussion is how we training structural biologists, but in fact when we train the students, we train also future researcher, and this is very important, probably more important, because then they have all the knowledge to learn other things. But what is important, it's one more time, it's the critical thinking, it's the scientific integrity, the ability to evaluate their data and to know if there is something wrong or not. And we mentioned also, so the prediction is very useful, very, it's under the experiment, but it's very fast now to work with this. But then you have to confirm that it's true, and then experimentation is necessary at the end. So we train future researcher, scientists.
Yeah, I totally agree. And there's a really good point about the problem solving, which obviously is absolutely critical. Yeah.
Melanie Fulmer from PDBE. So first of all, I want to make a comment, and that's for all of the younger participants or those early in career. Really consider programming. And I don't mean that you are a full stack developer who wants to work at Microsoft later on, but just the basic skills, because you make your own life so much easier when you have to write your PhD thesis up. Learn to do data analysis, some really good fundamental statistics, and then a little bit of programming to get those beautiful figures that go into your thesis, and you can repeat them all over if you realize you've made a mistake. Well, that's absolutely crucial. And then is something that I figured only fairly recently. I've had a number of years in my career, but only for the last couple of years I've had a mentor. And that has changed my perspective of how to approach my career. I thought it was all about the hard skills, you know, my knowledge, my being good at harvesting crystals and collecting data. But what I've never had was a mentor that actually puts me back from the scientific side and just consider what I want to achieve. And that has been absolutely marvelous to have someone on my side who gives me that. And that person is not my supervisor, is not my day-to-day advisor on the business side. And this is great. So can we set something up in our community that we can connect young researchers at the start of the career with a mentor who gives them an alternative view, not the day-to-day business and their hard science, but who gives them sort of a step back and gives them a guidance? That would be my question.
I think that would be an excellent idea, and it's definitely something to perhaps even for the organizers to write something about that in the sort of overview of the study weekend, because the study weekend is, of course, also to learn things, and these are not just the scientific skills, but really becoming a full-blown person. So that would be excellent, and I think that is a good point to end on, and we can go to the poster session and discuss it a bit more, because it's very interesting. So I thank the speakers.
Yep.